Okay. All right. So this PowerPoint is going to cover uh, material for carbohydrates. Typically, when I teach these classes, the first PowerPoint I just told you that I just went over with alcohols, that would last a class period in the fall, and this one is going to take at least two. So this is going to be a long PowerPoint. So it it could be 2 hours long. So that means is that what you should probably do is just break it into segments as you so decide to do, and I might give you points where you should do that. Maybe every 25-30 minutes you could do that, and I'll let you know. All right. So, uh, in the last PowerPoint, I said we had something called glyceraldehyde, and I told you that it's our first carbohydrate. Okay, and what our first carbohydrates have is they will all have at least the basic units will have the same simple chemical formula, CH2O, and we'll see how that relates to the name carbohydrates. And you'll also see is that they all have similarities, is that they have more than one hydroxyl and they either are aldehyde or hydroxide. So, welcome to our first BMM, biological macromolecule. I went on the internet and looked up carbohydrates, and this is what they gave me: potatoes, corn, bananas, bread, peas. Okay, guess what? Okay, these are all seeds. Okay, bananas. These are probably apricots, corn seeds, potato seeds. We'll see why. Okay. All right. So, but carbohydrates, another name for carbohydrates, not seeds. But that's where. All right, carbohydrate. All right, what I told you in the last slide on PowerPoint. This is a polyhydroxyl. So it says polyhydroxy. So polyhydroxyl aldehyde. Okay, or a ketone. So it's going to have a double bonded oxygen in it and hydroxyls. Okay, the name carbohydrate is actually pretty cool. So when you look at the word carbohydrate, it looks like it means carbon that's been hydrated. So carbon that has water attached to it. Well, if you go back and look at this, the last slide on the PowerPoint, the simple formula for glyceraldehyde was CH2O. CH2O. So, looks like it's a carbon that's been hydrated. Alright, and it turns out that this is common between all the building blocks of carbohydrates. They will all have the same simple formula. For instance, glucose is C6H12O6, that's its molecular formula, but its empirical formula is CH2O. Okay? So that's where the name carbohydrate comes from. Right, different classes of carbohydrates. The first class is called a monosaccharide. Let's go over this word. Mono means one. Saccharide means sugar in Greek. Okay. Now, it turns out that the other form of sugar is glue, okay? Like glucose, glycemic, or whatever, and that's the Roman derivative, okay? So one sugar, and it's the building block of eventually our macromolecules, polysaccharides. The types of the monosaccharides that we're going to be talking about in this class, besides the one we just talked about, glyceraldehyde, will be glucose, fructose, and galactose, and ribose and deoxyribose. Okay, now one of the things about naming sugars, okay, here my word sugar. So when you hear me say sugar, what do you should think? Carbohydrate, okay? Carbos, okay. So all the basic building units and even more, if it's a sugar, usually the name will end at "-ose", just like the alkanes and an ane, etc. So a triose is a sugar, okay, a monosaccharide, a building block, with only 3 carbons. A pentose, like pentagon, O sugar, so it's 5 carbon sugar. Hex, 6 carbon sugar. Okay, other classes will be disaccharides, which will be 2 monosaccharides linked together. Maltose, sucrose, and lactose will be the 3 that we look at. And then the really big macromolecules of carbohydrates, polysaccharides, which means many sugars. So these guys include starch, glycogen, and cellulose, which will be covered in the end. So the first thing we'll do is we're going to cover our monos, and then we'll talk about our dyes, and then eventually our polys. So here's this diagram. So a monosaccharide kind of looks like this, and we'll talk more about it, but it's monosaccharide. So saccharide again is Greek for sugar, 1 sugar, and then a disaccharide would be 2 of those guys connected, and a polysaccharide would be many. And so this might be like glucose, fructose, this might be like maltose, and then this might be like glycogen or starch. Alright, so when we discuss carbohydrates, we're going to end up talking about isomer. And isomer, remember, is same chemical formula but different bonding. Okay. 3 monosaccharides we'll be looking at. Okay, they're commonly found in our diet are glucose, fructose, and galactose. They always have the same chemical formula, but they're not the same. They don't have the same bondings. So they're isomers, which means same chemical formula, different bonding. Okay, so they're same but different. So glucose and galactose are almost exactly alike when you look at them. The difference is that they have one of the carbons is slightly different than the others, and it's how that carbon is situated. And the difference between these 2 guys is what we call these 2 guys stereoisomers. And basically what it means, stereoisomers, is that when you look at them, especially 2-dimensional, if I were to draw them 2-dimensionally, they would look exactly alike. But when you look at them in 3-dimensional space, you would see that they are different. And we'll see why in a second. Okay, so a kind of stereoisomer is something called a mirror image. So molecules that can have mirror images, and we'll see that in a second, are called enantiomers. Okay, so kind of fancy word here. Enantiomers. And they are mirror images of each other. And it's really hard to represent this in 2 dimensions. I'll try and give you the basic ideas, but the main thing for you guys is being able to identify if a molecule has a mirror image. For instance, an example of something that has a mirror image is your hands or your feet. If you took your right hand or your left hand and you put your right hand on top of your left hand, you'll see that they don't fit. Okay, you see that your right thumb is, your right hand's thumb is hanging off your left hand over on the side, and they're still alike. But if you put your right hand and put it in the mirror, all of a sudden you got your left hand. Okay, so your right hand is a mirror image. So in a sense, your right hand is an enantiomer, a mirror image of your left hand. And molecules are like that as well. Just like this diagram here. Okay, you have your left hand, you have right hand, and you can see they're different, but you put them in the mirror and it looks like they're the same. Okay, it turns out that most of the monosaccharides will have mirror images. And what makes the mirror image is this thing called chirocarbon, which I'll talk about in a minute. So glyceraldehyde will be our first example of that, which will show. Okay, so enantiomers, or molecules that have mirror images, what makes them have mirror images is this thing called chirocarbon. And so what we have to do is be able to look at a chemical, a bonding structure, and see if it has a chirocarbon. If it has a chirocarbon, it has a mirror image. If it doesn't, then no mirror image. Now it's really hard to show this in 2 dimensions. It's really hard to show. You got to play with the models. You got to get a model out, the molecular models, and look at it 3-dimensionally to see how it works. It's tough. I mean, it's not easy to see how this works. Okay, but what we want to do is we want to at least get a general idea of what's going on. So since we can't really see what's going on, we still would like to see if a molecule has a mirror image in it. And the way that it has mirror images is that if it has a chirocarbon. If it has a chirocarbon in at least one, okay, it could have more than one, then it will have a mirror image. That means that the same chemical formula can give you different structures. Now the way that we can tell is that if we look at a carbon, and if the carbon has 4 different things attached to it, then it will be a chirocarbon, and therefore it will have a mirror image. And it's important how we draw it. Now, notice the words here, 4, okay? Not 3 or 2. It has to have 4 different, and they have to be different. And this is going to be you have to be careful about how this, when we see it in a minute. It may not be real clear, but hopefully as we go through it, you'll see how it works out. Okay, so what we're going to do is we're going to look at glycerol. So if you remember, glycerol has 3 carbons, with each carbon having OH coming off of it. Okay. And again, the carbons have hydrogens, etc. Okay. And then we have glyceraldehyde. Where the top carbon is replaced by a carbonyl. Okay, so this, when we talked about in the last PowerPoint, this was the oxidized version of this guy here. So what we're going to do is look at this guy to see if if any of these carbons are chiral, and if they do, that means it has more than one structure. and then we'll do the same for glyceraldehyde. Right, so glycerol. So here's the structure of glycerol. What we can do is look at each carbon. So this is the first carbon here. We're going to look at the top carbon, and we're going to see if it has 4 different things attached to it. If it has 4 different things attached to it, then it's chiral, and then this structure has a mirror image. Well, you can see it has an H and H. That's the same. And now here's the thing I want you to see. This is that. So it has a carbon attached here, okay? But you have to look at this whole thing down here is a group, okay? This whole thing is a group. So this carbon has 2 of the same thing, so it doesn't have 4 different things. So it has three kinds of things attached to it. So this carbon is what we call achiral. So so far, this molecule does not have a mirror image. Okay, so let's go down to the second carbon, the middle carbon. And the middle carbon, let's look at the things attached to it. So it's an H and an OH. And then it has this group up here, and if you look at down here, it's the same thing. So the top and bottom are the same, so it doesn't have 4 different things attached to it, so this carbon is achiral as well. And then how about the last carbon? This last carbon has, just like the first one, has 2 H's attached to it. And remember, it has to have 4 different things attached to it, does it? So it doesn't matter how you draw this structure here. You could take this OH here, and we could put it over here, and it'd give me the same structure. So it doesn't matter how I draw this structure would always be the same. You could have an OH on the bottom here, I could have over here, I could have OH over here, and so forth. There's only one structure for glycerol. Okay, now let's look at glyceraldehyde. So let's start from the bottom. So this has 1 carbon, and notice that it has a hydrogen, so it has 2 of the same thing, so no, it doesn't have 4 different things attached to it. So this carbon here is achiral. Right, so now let's go up. Let's go to number 2. Well, let's be careful about this, okay? So it has H and OH. It has this thing, and it has this thing. So it has 4 different things attached. Now I know it has carbons attached, but the carbon has the different things attached to it, which it's a big deal, all right? So this is a chiral carbon. So this molecule here can have a mirror image, and the mirror image is based on this OH here, which we'll get in a minute. Sorry about the mess. All right, finally, let's look at the last one. Last carbon, just finish it off. So this carbon has 1, 2, it only has 3 things attached to it, so it doesn't qualify. But, so this overall, just because it has 1 chiral carbon, that means is that it can have at least 1 different structure that's a mirror image. And we'll show you that mirror image. Okay, now what I'm going to do is, if you go back and look at the last slide, is the bottom was represented as this right here. What I'm going to do is I'm going to replace it with this right here, okay? So you can see it has the same things. So it's CH2 OH, okay? It'll make things easier how we draw it. Right, so glyceraldehyde can have 2 versions. It can have and these are mirror images of each other. And the mirror images are around the chiral carbon. So putting the OH to the right is different than putting it to the left. Now if you put these in a mirror, they will look exactly the same. But if you put them on top of each other, they're like left hand on right hand. You can kind of see that. If I took this guy right here and plopped it on here, you can see that's over here. But if I put this in the mirror, of this guy, show it in the mirror, it would look just like it. Okay. So so we have to name the two. So what we decide is that if the OH goes to the right, we call it D, and that's like the German word or French word for right, dextra, and then L, levo, is left. Okay. So you're gonna have to be able to draw these two structures. Okay. So they both have the same tops and bottoms. The difference is in the middle. So you have to know how to do these, okay? Also, the numbering of these is from the top. So this is carbon number 1, number 2, and number 3. So the second carbon is the chirocarbon. Now, that's just for glyceraldehyde. Okay, so now it's why is this important? Okay. In life, the D version or the right version, okay, not because it's right, because it's right to the right, is the one that we prefer, our bodies prefer, okay. It's kind of interesting is that if you were to eat a carbohydrate with the left, it would taste like nothing, okay. But if you eat one with the right, it'd be very, very sweet, okay. Now, one thing we're going to find is we're going to go over 5. We're going to do 5 more monosaccharides, and all of them are going to have this in common at the bottom. All right, so what we're going to do is we're going to go, so you have the first 2 glyceraldehydes, which are trioses because they had 3 carbons. But there's 5 more that we're going to learn. There's 2 pentoses, that means they have 5 carbons, and there's 3 hexoses, 6 carbon. You're going to have to be able to draw all 7 of these. Yep. You're going to have to do that. Now, that's maybe sound a little intimidating, but if you pay attention, just like I said before, you look for the similarities and then you go for the differences, okay? So the similarities between all 7 of these guys includes the glyceraldehydes that we just showed, is they all have the same bottoms. Okay. Now, all of them are going to have the same tops, except for fructose. And then the difference is really going to be the middle carbons. So the top and bottoms are going to be similar. The middles are going to be different. So pay attention how we do this. Okay, so so let's go with pentoses first. So pent means 5. Os, sugar. There's 2 of them. D-ribose, and we're just going to always deal with the D, except for you're going to have to know L, glyceraldehyde, and deoxyribose. Okay, you probably heard of these before. RNA, ribonucleic acid. Okay, deoxyribonucleic acid. Where do they get this deoxyribo from? This deoxyribose. Okay. So what we're going to do is we're going to go through the structure of these 2 guys. And we're not going to talk about their function. We'll leave that when we get to the... Right, now the method to draw all the monosaccharides is this. First of all, what you do is you draw the correct number of carbons. So if, let's say you have a pentose, so you know that's 5. Okay. Next thing you do is that all of them have the same bottom. Okay, and this is the chiral carbon. The second carbon up is the chiral carbon. Right. The numbering of the carbons one from the top to the bottom. 2, 3, 4, 5. Okay. All right, and then the tops are all going to be the same except for fructose. See. Okay. All right. Okay. So this is the beginning of our pentose. We're not done, because now what you have to do is have to finish up these two guys right here. But that's the start. Is the bottoms and the tops are the same for all of them. Okay. The difference will be the middles, and we'll talk about the middles next. Right, so let's look at ribose. Okay, ribose has 5 carbons, like we just drew. Okay, here's the bottom, just like I brought, and here's the top. Deoxyribose, same thing. 5 carbons, same tops and bottoms. Okay, now what you have to do now is how you put the OHs. So the OHs, so you have to determine the middle OHs, how they go. Well, for ribose, both of them go to the right. Okay, right starts with RI, ribose starts with I, so both of these go to the right. Okay, now deoxyribose is the same as ribose, but what it's doing is it missing, deoxy means missing an oxygen, and it's missing the oxygen at number 2. So here it is, right here. Okay, so that's it. So 5 carbons, bottom, top, bottom, top, middles, right, difference, second carbon. There you go. That's what you have to do. Okay, now it turns out that those structures here were called linear structures. In nature, the structures are found different than linear. They're found in a circular, just like cyclohexane and so forth. And I'll show you these guys, and I'll tell you what you won't have to draw these. So there's a cyclo version of a ribose and deoxyribose. You won't have to draw them out. You just have to do a couple things. Number the carbons and tell me alpha, beta, which I'll tell you in a minute what that means. Okay, so what happens is the number 4 carbon connects with the number 1 carbon. And what it does, it forms this structure right here. And it turns out, if we look at this carbon here, the number 1 carbon is right here right now. Okay, if you look, it has 4 different things attached to it. It has H, it has an OH, it has an O, and it has a carbon attached. This thing attached to it. So this guy right here is a chiral carbon. So how this OH shows up, how we draw it, if it's on the bottom or the top, really makes a difference. Just like in our drawings, where this guy's going to the right or to the left. Okay, so what we do is, if this OH is going down, it's called alpha D ribose. We're not going to have L ribose. So here's what you're going to have to do with these structures. If you see this structure, you're going to have to number the carbons: 1, 2, 3, 4, 5. Okay, and so it goes clockwise, and we're going to, and usually it starts from this carbon here. So 1, 2, 3, 4, 5, and look at the last carbon is actually outside the ring. So it's not the carbon inside the ring. It's 1, 2, 3, 4, 5. Okay, so, and the other thing you have to do is tell me if it's alpha or beta. So if this, it's just this guy here, all right? It's not this one, this guy, not that one, that one, that one, that one. It's just this one here. And this one's pointing down, that's alpha. And then the next slide, you'll see that's pointing up, and it's beta. Okay, so here you go. So this is the beta version. It sounds like a computer program. So number 1 carbon, number 2, number 3, number 4, number 5. Okay. So this oxygen is not the 5th one. Okay. And the OH. Okay, not this OH here or this one here. This one over here that's attached to this carbon here is going up. So we call it beta. So beta up, alpha down. So that's what, so given a ring structure, what you're going to tell me, well, you have to tell me if it's a pentose or hexose. Here's it's a pentose, it has 5 carbons. You have to tell me number of the carbons, 1, 2, 3, 4, 5, 6, 5, I mean, and if it's alpha, beta. Now when I get to fructose, you have to be careful about it because it's going to be there might be a little kink thrown in there. And here's another version where we leave the carbons off, so we don't draw the carbons in. And you number them 1, 2, 3, 4, 5, and this is alpha pointing down. Okay, next thing, hexoses. So hexoses have 6 carbons, and there'll be 3 of them: glucose, fructose, and galactose. And we're just going to look at the D versions. So the second carbon go up with the OH will be pointing to the right. All these have the same chemical formula, but they're going to have different structures, and that's because they're enantiomers of each other. Okay. So let's look at glucose first. So glucose has 6 carbons. So 1, 2, 3, 4, 5, 6. So 1, 2, 3, 4, 5, 6. Here's the bottom. It has the same bottom everything before. This OH to the right because it's D, and has the same top. Okay. Galactose is the same. Has 1, 2, 3, 4, 5, 6 carbons. Bottom the same, top the same. All right. Differences in the middles. Glucose, the OHs go right, left, right. Okay. And for galactose, the OHs go right, left, left. And so the middle carbons is where they're different. So for glucose, you have R, right, left, right. And kind of a way to remember that is run, little rabbit. Run, little rabbit. And then for galactose, it could be run, little leopard or lamb or lion or whatever you want. Okay, lizard. Okay. Okay, so similarities, 6. Bottom and top. Difference, the middles. Run, little rabbit. Run, little lizard. Okay. So you're going to have to be able to draw these, just like you did the 2 pentoses. Okay, now let's look at fructose. Okay, fructose is almost like glucose. So go ahead and draw glucose. So it's 6 carbons. 1, 2, 3, 4, 5, 6. Here's the bottom, and here's the top. Then you got right, left, right. Forgot to put that guy in there. Okay, so run, little rabbit. Here's your glucose. Okay, now what we're going to do is we're going to flip the tops. So the top up here is going to go over here, and this guy's going to go over here. So they swap places, and now we have OH at the top and the carbonyl at the bottom. So now this is a ketone. Okay, these are aldehydes because the OH comes off the last carbon. The carbonyl comes off the last carbon. Here you have a trapped carbonyl. So this is a ketone. All right, and notice it's still right, left, right. Differences is that, and don't put another H over here. Okay, so remember, carbon has 4 bonds. So that's glucose. Okay, just like the pentoses, these structures can have ring structures as well. And we'll just look at glucose. What happens is the number 5 carbon will connect it with the number 1 carbon, like here, and then what we could do is have this drawing, these two drawings here, ring structures, okay, and you'll have alpha and beta versions as well. So what you're going to have to do is, if I draw this, you're going to have to number the carbons: 1, 2, 3, 4, 5. So each point in the ring is a carbon, except for this right here, and then 6. So number 6 is outside the carbon, and this is alpha pointing down. Right. So number of carbons: 1, 2, 3, 4, 5, 6. Pay attention. Okay. And then this is pointing up, so it's beta. Okay, you may want to take a break at this point and finish about halfway through. Okay, so here's the last one. Here's the numbering: 1, 2, 3, 4, 5, 6. Here's the last carbon. Each point here is carbon, 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 carbon. All right, fructose. So fructose is going to be a little bit different. Okay, it's not going to look, even though it has 6 carbons, it's not going to have a shape that looks like 6. So for galactose and glucose, we saw they had 6 ring structures. But remember, one of the so the carbons went 1, 2, 3, 4, 5, 6. Okay, the oxygen is not part of it. What we're going to see here for fructose, it's going to be 5 ring structure. So fructose 5. Okay, and what it does is it has 2 carbons coming off the chain. So it's 1. So the first carbon, this is number 1 carbon, not the one that's in the chain. So 1, 2, 3, 4, 5, 6. And we'll see that next drawing. So if this seems messy, then you'll see how it applies in the next drawing. So here's fructose in the ring structure. Again, you have to number and tell them it's alpha or beta. So this is the number 1 carbon. 1, 2, 3, 4, 5, 6. So notice that the first carbon is not in the ring. Okay. This OH is one that determines the alpha or beta. Pointing down, alpha. Now, one thing you may notice, I don't know if you would or not, but I'm going to show you, is that fructose actually looks a lot like ribose. They both have the same structure, and they both have this 5 ring structure. But what the difference is that fructose has a carbon instead of a hydrogen here, up here. Okay. So it's not the shape of the structure that tells me if it's a pentose. Okay, so this is 5-ring structure, but it's a hexose. It's the number of carbons. So this has 1, 2, 1, 2, 3, 4, 5, 6 carbons, so it's hexose. It's not shape that determines it. All right. Now what we're going to do is we're going to talk about the function of our 3 macromolecules. And this is what we do is going to talk about glucose, D-glucose. All right. D-glucose is found in all living things. Plants, animals, bacteria, etc. Okay. In animals, we often store it as something called glycogen, which we'll talk about, and often stored as starch in plants. But it can also just float around. It's the super molecule of our bodies, okay? Super mighty mouse it is, okay? Don't forget that. It's now the previous teacher that gave me these PowerPoints told me that glucose was just found in grape and honey and things like that. That's all wrong. Glucose is found in every single living cell there is, because every single living cell requires, well, doesn't require, but prefers glucose as an energy source. So if you were looking at just any cell in the universe, on this earth, you would find glucose in it. So if you were to take some hamburger and you were looking at, open up the cells and look at the hamburger, you would find glucose in it. Okay. If you were to take a plant and smash it up and look at all the cells, you would find that the main molecule that shows up in it is glucose. Glucose is, what we just told you, is a monosaccharide, is based biologically, is the main source of metabolic energy of all living things, okay? It is the main energy source of all living things. Now, humans and some other are able to use fatty acids, but almost all living things prefer glucose as their energy source. This is when you're out there, when you go in the nursing field, and they'll talk about sugar. The sugar that they'll be talking about is glucose. It is the energy source. It is what gives your brain, your cells energy. Okay, I can't say enough about it. You really got to know this. Okay, when you take, we'll see as I go through these. Now, all other monosaccharides have tiny bits of use, but they're important uses, but not like glucose. All right, so let's just go over this. Where do we get glucose from? So glucose, well, where we get glucose, and this is kind of cool stuff, is that there's this process called photosynthesis. And if you have biology, you've probably heard of it before. But what happens is that plants do this. Okay, some other things do it as well. But they make glucose. And it says make carbohydrates. And so, you start hearing all these terms. You hear carbohydrate, glucose, right? So when it says make carbohydrates, specifically what it says it means is making glucose, which is a type of carbohydrate, which I might call a sugar. And what it does is it takes the carbon dioxide from the air, which is really cool, and it takes water from the ground, and it combines them with sunlight, in the cellular mechanism of these plants, and it makes glucose. And the other thing it makes is oxygen. Cool. Okay. And so the plants then, what they do is they make the glucose, and what they do is they store the glucose so they can use that glucose to make help them do the things they need to do. Okay, so we're going to actually go through the chemical reactions here. So photosynthesis, it turns out that cells are pretty amazing things, but what they do is they take a very simple chemical reaction and they make it very complex to make things simple. All right, seems weird, but so what happens in plant cells is they take energy from the sun, they take carbon dioxide from the air, combined with the water and the ground, with a bunch of stuff here, sunlight, enzymes, chlorophyll, blah blah blah, and it makes glucose and carbon dioxide. I mean, oxygen. So plants make glucose for their own energy, so they're called autotrophs. Animals, by the way, are called heterotrophs. That means is they don't make their own energy; have to get their energy from other places. Why do plants make glucose? Well, they don't make glucose for us. What they do is they make it for germination, so that so their seeds can grow. And also, we don't think about it. Even though plants just sit there and look like they don't do much, they still are living and need energy. And and where do they get the energy to do their work is from the glucose that they get from making the glucose. Animals, what we do is we take the glucose. So we're kind of what do they call it? We take advantage of plants, and we steal their glucose, and we eat it. And what we do is when we eat the glucose the plants make, it produces energy for us to do work. And this process is called cellular respiration. Cellular respiration has many steps, but the overall chemical reaction is that living things, animals, not just living things, animals will eat glucose, and it could be, a primary place we can get it from plants, but we can get it from other sources, is like cows. When cows, I don't know, like I said, every single living cell there has glucose in it. So if you eat things with it, eat a cell, you're gonna have glucose. But where did those cells originally get the glucose come from? It was plants. So glucose. So when we take glucose in, and we'll be more specific in a minute of this, we breathe in oxygen, we give off carbon dioxide and water, and it produces energy for us to do work. And if you notice, this chemical reaction is the opposite of photosynthesis. So that's why we need plants, because plants and animals do the reverse chemical reactions. So here's photosynthesis. It takes energy from the sun, combines it with carbon dioxide and water, forms glucose and oxygen. And then what we do is we do the reverse. We take the glucose, we combine it with the oxygen that the plants give off, form carbon dioxide and water and energy. And guess what? The carbon dioxide goes back to the plants and the water goes back to plants. And we go, and this is a cycle. And it's called the carbon cycle. And here's a pretty picture of it. So you have the plants over here, And what they're doing is they're taking in carbon dioxide and water and combine with the sun, and they're making glucose. The horse breathes in the oxygen, eats the carbohydrates, and produces carbon dioxide and water, and goes off running really fast because now it has energy. Okay, here's another version of it. So you have the sunlight combines with carbon dioxide and water forming glucose and oxygen gas. Okay, well, those get put into cellular respiration, which takes place in the mitochondria. Combines with the oxygen you breathe, forms carbon dioxide and water, and those two things go back in to plants. And so what you get is plants give animals glucose and oxygen, and we give the plants back carbon dioxide and water. So it's a reciprocal living relationship between plants and animals. Right, so when we eat glucose, again, it could be more than just plant. Like I said, you could have your hamburger or anything with any cells. All cells probably have glucose in them. So what happens is the glucose goes into your stomach and into your gut. And what we want to do is watch what happens to the glucose. The glucose gets passed from your gut into your blood. Okay, at this point, the glucose in your blood is called blood sugar. Okay, you probably heard of that. Okay, but it's just glucose. All right. So your blood has glucose in it, and we call that blood sugar just because it's in your blood, and it's sugar, it's glucose. Right. Eventually it gets to the organs, your muscles, your brains, etc. And then what happens is that it tries to get into the cells. Okay, now there's a problem here, is that the cells just don't let anybody in. There has to be an invitation to come in to the cell. So what happens is that when this blood sugar, as the level of glucose gets higher and higher in the blood, it sends a signal to the brain. It says, we have food sugar and we need to have it put into your cells. And what happens is the brain tells the pancreas to release insulin. And the insulin is released, and what the insulin does is it goes and opens a door for the glucose to get in to the cell. And then what you do is it undergoes cellular respiration, which we'll talk about. So that's what happens to the sugar in your blood. Now let's talk a little bit about that. So, all right, your body has many organs, and all those organs need energy. And where they get the energy from is glucose. Now, the most energy using organ in our bodies is the brain. The brain is continuously just working all the time, even when you're asleep. It's working. And for it to work, what it needs is energy. and where does it get energy? It needs glucose. So the brain continually needs a influx of glucose. All right. So okay, now the other organs really don't need that total influx of glucose, because most organs have ways of storing it, which I'll talk about a little bit. So yeah, I don't know if you ever experienced it, but if you're really low in blood sugar, your brain will start to feel kind of woozy and you might pass out. Okay. And so it's kind of important thing you need to do here. All right. Now, all the organs need glucose as well, but not as much as the brain. The brain continually needs glucose pumped into it. Normally, the glucose is in the bloodstream, and the way we eat these days, we really don't need to put a whole lot of glucose in our systems. There's always enough for our brain, and I'll talk about what happens when our blood sugar goes low, how our body adapts to that. But typically, the concentration of glucose in our body are 70 and 90 milligram per deciliter, which is about 100 milliliters, and it'll go up after eating, and we'll show that in a minute. And if you go into med tech field or nursing, you'll see that these numbers are pretty important. Okay. High levels of glucose or presence of glucose in urine maybe indicate that you have diabetes. So let's say you've been feeling tired, and you've been drinking a lot of water recently, and you just don't feel yourself. Okay, so you go to the doctor, and the doctor says, you know what? That kind of sounds like you may have have diabetes. And what they'll do is that they'll have you take a urine test, and then a couple other tests, and they'll take urine tests to see if you have high ketones, or your body's undergoing ketosis. That the reason why your body would be undergoing ketosis is because it's can't use sugar, it has to use fat as the energy resource. And if you were in my class, I would spend more time talk about. Hopefully, you'll learn about this in A&P more. But so let's say you go to the doctor, and they say what they do is they give you a test, and there's different tests they have now. They call A1C, but the other one they have now is this glucose testing. And so what they do is they give you some glucose, and what they do is they measure your glucose in your blood. And let's show you what that might look like. So let's say they give you the glucose at 0 time here, and these are 3 possibilities that you could have. Normally what would happen is the glucose is going to go into your blood, and it's going to increase and then it's right about here is when the brain says release insulin to have it go in the cell, and then the amount of glucose disappears from the blood because it goes into the cells and it drops off to normal. Okay, now if your body doesn't make insulin, what happens is so your blood levels go up, but so now you don't have there's no insulin to let this glucose into the cells, and so your glucose levels stay pretty high. Now they do go down, And that's because there's other ways that the glucose can get into your cells. Okay, so hyperglycemia means high sugar levels. Remember at the beginning of this I said glyc is the Roman version of sugar. So hyper means high, sugar, and just systematic. Okay. Right. Now the opposite happens down here is that when you take it, it gets into your blood, and then your body just sucks it up, and it actually takes the glucose out of your blood completely, and which is not a good thing, because you need to have a certain amount of glucose in your blood to keep your brain working all the time. If the glucose levels go down too low, there's not enough glucose to keep your brain working. All right, so hyper could mean that you have type 1 or type 2 diabetes, and there's a difference. Type 1 is typical, it's called juvenile, kind of born with it, or it happens in youth, and type 2 is adult, and it kind of happens because of your environment and the way you live. Hypoglycemia is also hypoglycemic. Some people would have this with low blood sugar. Right, and here you go. Okay, so now let's talk about what happens when the glucose gets into your cells. How does it actually produce energy? Okay, and it turns out that the overall chemical reaction is really simple. It's glucose, C6H12O6 plus O2, I'm going to leave off how many, yields carbon dioxide and water and energy. Okay. So that's what happens overall. Okay, but it doesn't do it in one simple step. Okay, that, like I said, nature makes things complex to make things simple. All right, so what happens is this: the glucose goes into your mitochondria, and what happens is this first step called glycolysis. So there's going to be 3 steps here. And the first step is to break the glucose into 2 smaller units. And you don't have to draw. Just know that it splits it into 2 smaller carbons, right? Okay, so that's glycolysis. Notice nothing's lost or gained during this process. It just splits it. Okay, next thing that happens is that this guy here gets put into the next step called the Krebs cycle, which I'll talk about in a second. And during this process is that carbon dioxide is released right here. Okay. And the hydrogens get shipped for the next step. So the first step is glycolysis, splits it in 2, and the Krebs cycle releases the carbon dioxide. So all the carbons and the oxygens are gone, and the only thing left are the hydrogen. And you don't have to get the numbers here; you just have to get who's doing what. And the next step is called oxidative phosphorylation, and this is where the oxygen that you breathe combines with the hydrogen, and they form water and energy. Okay, and if you look, the ends are glucose and oxygen, and the outs are carbon dioxide and water and energy, exactly as this chemical reaction up here. And then the energy goes and makes ATPs, and the ATPs can be used to make energy, to make energy, muscles move, and etc. Okay. So one of the cool things about this is that when you breathe in oxygen and breathe out carbon dioxide, you may think that the oxygen you're breathing is being released, coming back out as carbon dioxide, but it's not. Okay, the oxygen that gets the carbon dioxide that you're breathing, the oxygen that you're breathing is actually coming from the glucose that you eat. So the glucose that you have eaten is where the oxygen and carbon dioxide comes from. The oxygen that you breathe actually is part of the water that's given off. Where's that water? It's like sweat and other things. Okay. So you have to know these three things. So there's glycolysis, which breaks glucose into 2,3 carbohydrates. That's all you need to know. Then the 2,3 carbons go to Krebs cycles, and they release carbon dioxide out of the cell. And then the hydrogens get moved on to the next step, oxidative phosphorylation. They combine with the oxygen we breathe, forms water and energy to make ATP for us to be able to do work. Okay. All right, at this point, you probably want to take a little break, and we'll be finishing up the monosaccharides and get to polysaccharides in the next. We'll be done in the next half hour. Okay, so we talked about glucose. That's that monosaccharide. Now we're going to talk about the other two monosaccharides we talked about, D-galactose and D-fructose. We're going to talk about their functions. Okay, we talked about we went over their structures. Now we'll go over their functions. Like I said at the beginning of this class, what we'll do is we'll go over structure and then function. Okay, so galactose is found in lactose, which is milk. So milk, we're talking about milk here, we're talking about mammal milk, not like soy milk and almond milk and all that fake milk. We're talking about milk, okay? Milk has galactose in it. And we'll see in a second how the lactose is actually stored in a molecule called lactose, which we'll get into a little bit. So D-galactose is important in cellular membranes, especially for young people. And if if you remember, the difference between galactose and glucose was this was Run Little Leopard, and this is Run Little Rabbit. Okay, what happens? There's this enzyme called epimerase, which changes the. Now, if you don't have that enzyme, it turns a lizard into a rabbit. Okay. Then what happens is that you'll get a buildup of the D-galactose, and that can cause serious problems. Okay, and one way to make sure that doesn't happen is to just make sure you don't eat milk products. And these days you will know this through genetics and parents and knowing about your parents' background. So galactosemia. Okay, this is not lactose intolerance, which we'll talk about in a little bit. All right, D-fructose. Okay, what is its function? Oh, well, yeah, what is its function? Well, well, it comes. There's plants that have it. Okay, like corn and sugar and things like that. But the reason there's no real reason why we need it. Okay, there's no reason at all that we need fructose. The only reason why why we eat fructose is because it's sweet. It's about twice as sweet as table sugar, which I'll show you in a minute. Um, when you eat glucose, your body can turn it into glucose, but it's fairly difficult. It's almost kind of like how your body turns ethanol into acetaldehyde. Okay, it's a pain. Okay, fructose. There's been a lot of implications that fructose can cause all kinds of problems. Might lead to type 2 diabetes and stuff like that. So if you go and look at most energy drinks, they'll say that it has high high fructose corn syrup. I would strongly suggest staying away from it. I don't know. It's hard for the body to break this down, and when it does, it breaks it into fat, and could lead into all kinds of problems with it. Here's the kind of curious thing about it. Here's a sweet scale. All right, so this is an arbitrary scale where sucrose is given the value of 100. Sucrose is table sugar. Now, if we look at fructose is 175. So fructose, one molecule of fructose is about twice as sweet as sugar. So I mean, food manufacturers know that. So we crave sweet stuff. They put this in it, and it's cheap, and you get twice as much sugar, sweetness out of it. All right. Here's the curious thing. If you look at artificial sweeteners like aspartame or NutraSweet, it has a score of 18,000, which is 180 times sweeter than table sugar. Okay, so one molecule of aspartame is equal to like 180 sucrose. So that's why it's really powerful in terms of its sweetness. Okay. Okay, next thing we're going to get into disaccharides. So these are going to be 2 monosaccharides. And there's 3 of them that we'll cover: maltose, lactose, and sucrose. We'll go over their structures and their function. All of these are taking 2 monosaccharides and putting them together to make a disaccharide. We'll go over each one of them now. Okay, so here's the 3. So maltose is made out of 2 glucoses. Lactose is made of glucose and galactose. And sucrose is made of glucose and fructose. Again, notice they all end in "-ose" to tell you they're carbohydrates, sugars. And we're going to go through each one of their structures. And then we're going to go... Alright, okay, first thing I want to talk about is what links the 2 monosaccharides together. What links them together is something called glycosidic bonds. And there'll be different types, and I'll show you those in a little bit. So the first one is maltose. So maltose is also known as malt sugar. Is found primarily in germinating grain. We'll talk about that. and you may have heard of malt, like it's found in cereals, like malt drinks. And a lot of people heard of it in terms of beer, malt beverages, and things like that. But so maltose is simply the disaccharide that's found in the seed of germinating plants. And it's like this. So you have here, there's two glucoses. Here's a glucose and here's another glucose. Then what happens is that, and when you number them, so this is number 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. I goofed up. This should be up here. 5, 6. So ignore that right there. Okay. So the fourth carbon, and then combines with the first carbon here. So 1, 2, 3, 4, 5, 6. So first carbon combines with the fourth carbon, and they form a 1,4 glycosidic bond that points down. And if you go back to the when we talked about this earlier, when the OH points down, the O points down as alpha. So it's alpha 1,4 because it's number 4 linked to number 1. 1,4 alpha. Okay. So there's your maltose. So maltose has 2 glucoses. The oxygen points down, alpha 1,4 glycosidic bond. All right, and here's another diagram of it. 1, 2, 3, 4 combines with 1. Energy bond points down, so it's alpha 1,4 glycosidic bond. Okay, now when we eat maltose, is that we have an enzyme in our body called maltase in our guts, and what it does is it'll break apart the 2 glucoses from each other, and then you'll end up having glucose, and these 2 glucoses can go out in your blood and supply you with energy. Okay, lactose. Okay, lactose is milk sugar, which we talked about when we talk about galactose. It's combined. Lactose is a combination of glucose and galactose, and we'll talk about this in a second here. Lactose intolerance. All right, so lactose looks like this. It looks a lot like maltose, but instead it has a galactose and a glucose. And now the oxygen is pointing up, so it's beta. But it's the same thing, 1,4. Okay, beta 1,4. Galactose, glucose. And there's an enzyme that can break this apart into galactose and glucose. Okay, if you don't have this enzyme or lacking or it's low, then you can't break this apart, and that's what causes lactose intolerance. So if you have low levels of lactase, that will cause gastrointestinal irritation. Okay. And it's a genetic disease. So, and there's people around the world, different peoples around the world that have this. And so this is a little bit background behind it. Okay, now sucrose. Okay, sucrose is also known as table sugar. We get it from sugar cane or sugar beets. What it comes from is glucose and fructose make it up. All right, so here's what happens with it. So here's glucose and here's fructose, and they combine together to form this guy over here. Okay, and we'll take a look at this, but notice this is that the number 1 carbon combines with the number 2 carbon of fructose, so it's called 1,2. It goes down, which is alpha, and goes up, which is beta, so it's called alpha beta, 1,2 glycosidic bond. And here it is again. So on top is the glucose, on the bottom is fructose, and you get this connection 1, 2, alpha, beta. Okay, and there's an enzyme called sucrase, which will break this glucose and fructose into its components, glucose and fructose. Okay, now the final part of this PowerPoint and this section. Okay, so this is complete material for this test. Is the first PowerPoint. It's had to do with hydroxyls and carbonyls, and now carbohydrates. So finally, talk about polysaccharides. Poly means many, saccharide means sugar. We're going to talk about 3 of them, okay? And they all what they all have in common is they're all made of glucose, and there's 3 of them: starch, glycogen, and cellulose. We're going to talk about starch first. Okay, starch is found in plants. And we'll talk about why it's stored in plants in a minute. But there are 2 kinds of starch molecules. There's something called amylose and amylopectin. Amylose makes up less, and amylopectin will take up more. The amylose is going to be straight chain, and the amylopectin will be branched. I'll show you in a minute. Okay, so here's the amylopectin, more of it, it's branched and so forth. And so you don't have to really worry about this stuff here. Let's go into it now. Okay, so starch, there's 2 kinds of starches. There's amylose, okay, and here's an example of it. So the amylose, if you look at each one of these, is a glucose. Okay, and they all go in a chain, okay, a linear chain. Right, now the amylopectin, if you notice, is this guy right here, is that you can see that they're connected all over the place, okay? So there isn't just a straight chain with guys coming around like that, and so forth, okay? And the reason why this is that this is a really great way of storing a lot of glucoses in a very small space. This doesn't, okay? So let's talk about starch and how plants utilize it, okay? So you have your plant. A plant undergoes photosynthesis and it makes glucose. Now some of the glucose goes on to do the daily living of a plant, which isn't a lot, okay? Basically just keep it from falling down and wilting, okay? But still needs energy, all right? Okay, but what happens is a lot of the glucose gets put into the seeds of the plant, okay? And what happens is this glucose gets stored as glucose. I mean, as starch. So the glucose goes into the seed, and what this seed does is it combines all the glucoses together to form starch. Okay. Now, when a seed says, I want to germinate, okay, well, it doesn't have sunlight to give it energy, so what it does, because it's buried in the ground, is it sort of signals that tell the seed to start breaking down the glucose. And when that happens, the glucoses are broken off, okay, and from the starch, and what they do is the plants breathe, the seed breathes, it takes in oxygen, and what does it do is it makes carbon dioxide and water and energy to grow. Okay. So that's why plants make starch. Starch is the energy for the seed to grow. Okay, so what we do is we take seeds of these plants, and they're rich in glucoses because it's stored as starch, and we eat them. And when we eat the starch, it breaks it down to the glucoses, and the glucoses will give us energy. Okay, so that's how that works. So plants don't make us food for us; they make it mostly for their seeds to grow. Right, now the big guy. Okay, this is the most important thing here. Besides glucose, all right, glycogen. I don't know if you guys have ever heard of it, and I don't know if you ever hear it. Hopefully you hear it again. I hope your nursing people and etc. will talk more about it. But glycogen serves as the body's reserve supply of carbohydrates, glucose. Okay. So instead of storing it in seeds, we store our glycogen in our livers and our muscles and other organs. And we'll talk about more specifically this in a second. Okay. What it does is that when our body needs energy, just like seeds, it will break the glucose off of the glycogen and release it to produce energy. Okay, so let's talk about this process. Okay. Right, so let's say you have some glucose floating around your blood, and let's say it gets kind of high, and you don't have any problems with it. Your body says, well, I don't need any more glucose from my cells or my brain, so can you do something with the glucose? We don't want to just get rid of it. So as long as there's enough glucose in the blood for the brain, the extra glucose will go to your liver, and then what it'll do is it'll package it as glycogen. Right, and what it does is it's just like starch. It combines the glucose molecules together, kind of like amylopectin, to form this giant macromolecule with lots and lots of glucoses in it. Okay. All right. Okay. Now let's say that for some reason your blood sugar goes low, and your brain goes, help, I need sugar. Okay, glucose, that's what I mean by sugar. And well, you could go eat, but your body has a more efficient way of doing it. It says, don't worry about it. What I'll do is I'll crack off some glucoses off of this glycogen. So what it's going to do is it's going to break the glucoses off the glycogen. And what it's going to do is it's going to put it in your blood. The glucose will go to your blood, and then what it'll do is it go to your brain or other organs, and then undergo cellular respiration. Okay. So the liver is kind of sort of like a backup of glucose for your to make sure that you have enough glucose in your blood to supply for your brain and other organs. All right, now a problem comes with this. Is now if you exercise, or not even exercise, just live, okay, like muscles, okay, muscles require a lot of energy to work. Especially, I mean, if you evolutionary wise, human beings used to work and move a lot, and so they would use their muscles a lot, which means is that they would use a lot of glucose. Well, the problem is that where does the muscles get the glucose? If the muscle gets the glucose from the blood, let's say this way, what happens is that as the muscles start to work, what they'll do is they'll start shunting the glucoses to the muscles more and more and more, and then there'll be less of the glucose to go to their brain, other organs. Okay, all right, and then eventually the brain and other organs won't have enough energy to do what they need to do, and then you pass out and possibly could die. All right, so what our bodies have done to stop that from happening is that the muscles have their own source of glycogen. So instead of stealing the glucose from the blood to have it go to the muscles, it has its own glycogen, which releases glucose and undergoes cellular respiration. Okay, finally, the last guy is cellulose. Okay, like starch and glycogen, it's made out of glucose. Okay, we can digest starch. Our body can break apart starch. Our body can break apart glycogen. But one thing we can't do is we can't break apart cellulose. Okay, where is cellulose found? Okay, it's found in paper. Okay, trees, bark, cotton. Okay, so our bodies can't break this right here and release the glucoses to do energy. So it's a shame, okay? So if you have a piece of paper is made of thousands and thousands of glucose molecules, which are just potential tons and tons of energy. But we can't get the glucose, we can't break the glucoses off of the cellulose. We don't have the enzyme to do that. Okay? So if we did, we could solve the world's hunger problems. People would just go up to a tree and start eating it, right? And they get all the food you want. Because tree bark has thousands and thousands of glucoses in them, but we can't get them because we can't break apart. Well, there turns out there's species that actually can do this, like termites. Termites can eat cellulose. Take cellulose, break apart the glucoses from the cellulose, use that for energy. So if there's some way that we could take what the termites have and use it in our body, we could solve the world's hunger problems. But there is some use for cellulose. Even though we can't digest it, it's still good for our diet because it helps us move our bowels, okay, and have good things for our pooping, okay. Um, and so so this finishes up the this material for test two, and you're probably gonna you want to break this into parts. Um, like I said, I'll send you not a worksheet. I'll have a worksheet that kind of goes over, ask you questions about things, but just to make sure that you're on the right path. Uh, but it's up to you to look at the PowerPoints. Again, I will post the PowerPoints on the announcements and the non-voice and the sideboards. And this is turns out about an hour and 36 minutes. Okay, hopefully everything turns out fine, and end this lecture on the word poop. But no, so the next so that's it for this. We'll be done, probably taking the test for this by the end of I don't know, June 20th or somewhere around there. Okay, so that's it, and oh, okay.